Hi everyone, thanks for checking out this video. So there's going to be probably another quick lesson. I'm just going to show you guys how to make a heat map plot of our differential expression results. And this is a good plot to know how to make because this is one of the more common plots that you'll see in like papers that's used to communicate results like this. And also like just generally a good skill to have in your skill set because being good at data visualization it's like really important as like a bioinformatician and like a data analyst in general, basically. And knowing how to make like good plots to communicate results is one of the things that will make you like useful to experimental biologists and potentially like useful even as an employee who they would want to like pay to do this. There's definitely like data visualization is like one of the most important skills in this field to be able to have. And these plots, these heat map plots, are one of the most common types of plots for communicating like gene expression results, basically. So yeah, it's good to know how to make them, and it's also good to know how to interpret them if you see them like in a paper, for example. So I'll just start by showing you guys what the end result will look like. So this is what the heat map result is gonna look like, basically. So it has this like grid, like this matrix, where every row in this grid is represents like one gene, and they're labeled on the side here. And then every column represents one sample, basically. And if you zoom in far enough, you can see the samples like actually labeled according to if they were IPF or control samples. So it's a little bit like the actual like expression matrix. But yeah, it only has our differentially expressed genes, our like 79 differentially expressed genes. And again, this is part of why I used a stricter threshold than I probably would have used in real life, was just because I didn't want to have too many genes in this plot. I wanted us to be able to like actually be able to read this plot and see the labels on the side. But yeah, so we have a row for each differentially expressed gene and a column for every sample. And then each like square in this grid tells us how high or low the expression was in that sample for that gene, basically. So yeah, it's like red means higher expression and blue means lower expression. That's why it's called like a heat map, representing like the heat or the coldness based on how high or low that gene was expressed in that sample. So yeah, again, every square here is telling us based on how red or blue it is, how high or low that gene was expressed in that sample, basically. And then besides that, you can see there's some clustering going on. There's this like hierarchical clustering going on in this heat map, where the like both the genes and the samples are clustered based on how similar they are to like the other genes or the other samples. So there's this like bracket clustering, like grouping them together based on similarity for both the genes, which you can see the brackets here, and then the samples, which you can see the brackets here. And if you just look at this, we can see that like clearly there's something different about like the left side and the right side of this whole plot. So the left side is a lot of like gray, actually, a lot of like gray. And what that means is that the expression is like close to zero. I should say also, this is relative expression compared to the mean of the control samples. So like something that's gray or close to zero means that for that sample's expression was close to the mean expression of the control samples for that gene. And like the blue color means that a sample and a gene had like lower expression compared to the mean of the controls for that gene. And then red means higher expression in that sample compared to the mean of the controls for that gene. So yeah, the left side is a lot of grays. Whereas the right side, we have like two groups of genes, where this bottom group tends to be a lot of blue. So a lot of like lower expression compared to the mean of the controls for these genes. And then like the upper part of this is a lot of reds, meaning this selection of genes tended to have like higher expression in these samples on the right compared to the means of the controls. So before I tell you guys, I want you to like maybe pause and make a guess about what that tells us about like these samples and these samples. Okay, so I'll tell you guys that, and again, this is partly because we picked the genes like that were differentially expressed, so kind of knowing that this would happen. But yeah, these genes on, these samples on the right are the IPF samples mostly. Although there are some exceptions. See, like over here, you can see there's like some exceptions where there are some control samples that did have expression profiles across these genes that seem to kind of match the like IPF samples. But yeah, these are mostly IPF samples over here. And then on the left side, these are mostly control samples. And again, that didn't happen by accident. That is like to be expected because we chose these genes based on that being true. We purposely picked out the genes that had a difference in expression in the IPF samples like compared to the control samples. Yeah, so we purposely picked these differentially expressed genes, and then the result of that is that if we cluster the samples like this, then it'll tend to be that the IPF samples will tend to cluster together and the control samples will tend to cluster together. Plotting it like this also is a way to like possibly look for some outliers, like visually speaking, in terms of both the samples and the genes. So we can see down here like these genes, these bottom two, tended to have, even for the control samples, they tended to have like a lot of control samples having lower expression than the control mean, like by kind of a lot. So they're not just like all grays like we would kind of expect like up here. Yeah, these bottom two genes, even most of the control samples had like lower expression compared to the control means, like by kind of a lot. These ones are pretty dark blue. So we might ask, like we might then, if this was a real analysis, go and like look at these genes and see if maybe like a couple of the control samples, or even like one of them, was like an extreme outlier for having like very high expression in a way that like threw off the mean of the control samples, or something like that. Like this is like a way to like visually try to identify any genes we might want to look into as being like strange outliers or something. But yeah, that's the idea with the gene expression heat map plot. Yeah, you'll see this in a lot of papers used to communicate like results of like differential expression analyses. And yeah, it's good to know how to be able to interpret them. And yeah, now I'll show you guys how to actually make them. So what we need to do to make this basically is to come up with like a matrix of gene expression that we can use to pass into the Seaborn library to actually make this plot. But what we need to do is we need to standardize the measurement of high or low expression like across different genes, because like the CPM measurements for these genes could be like very different, but we want to like standardize so that we can use the same color scale for all the different genes. So actually what we're going to do again is this like log2 fold change to like standardize the like the gene expression being like high or low compared to the mean of the control samples. But this time, rather than doing like the log2 of the IPF mean over the control mean, we're going to do the log2 of each particular sample for a gene. Yeah, like each sample, including all the individual control samples, and then we're going to do the log2 of each sample measurement divided by the control mean, so that we can get a log2 fold change value for like each box in this grid, each like square here, including the control samples basically. So yeah, we don't just want the log2 of the IPF mean over the control mean. We want the log2 of each particular sample measurement for a gene over the control mean for that gene, basically. So yeah, like with that being said, after that explanation, the actual code to do this is pretty easy. We're going to start by just like making a NumPy array, two-dimensional NumPy array with the same shape of our significant gene expression matrix, which we called sig data, which has rows for the 79 differentially expressed genes and columns for the 206 samples, basically. And then what we're going to do is just basically go through all of our genes, and then for each gene, we're going to calculate the mean of the control samples for that gene, and then set our corresponding row in this like empty, or I mean not empty, but like full of zeros matrix we just defined. We're going to set the corresponding row to be the log2 of this ratio of like each sample's expression measurement in sig data over the control mean, but again doing this epsilon trick of like adding like a small constant, which will be 1 to both the numerator and denominator, yes, to both the top and bottom of the fraction, to avoid any like division by 0 errors or like log2 of 0 errors. And yeah, it's basically all it takes. And then after doing that, yeah, we can just use this Seaborn library to make a make like a nice heat map for us. So yeah, we can both save it as a PDF as well as display it in this notebook. And yes, that's basically all it takes to make this heat map. Yeah, so pretty easy, right? Yeah, so just again, just like a good way to visualize these results, basically. Yeah, so hopefully I explained this okay. Hopefully all this all like made sense to you guys. But if there was any confusion, if anything about this heat map plot or how we made it didn't make sense, please just let me know in the comments, and I'll be happy to try to answer any questions that you guys have. Okay, so that's all I have for this video. So thanks for watching, and see you guys next time.